So usually when we think of a baby being born, we kind of just assume the biological construction of the brain is, well, pretty much finished, right?
Right, yeah, like the house is fully built.
Exactly. The traditional textbook narrative treats it exactly like building a house. Like, first you pour the foundation, then you frame the walls, and by the time the baby is actually out in the world, the heavy lifting is done.
Yeah, the assumption has long been that the massive migration of neurons, the literal physical movement of brain cells to their final destinations, was securely wrapped up inside the womb.
But I mean, what if I told you that right now, in the brains of newborns all over the world, millions of neurons are actually packing their bags and embarking on this massive cross-brain journey?
It's honestly mind-blowing.
It really is. So today, you are joining us for a deep dive into a groundbreaking paper from Nature Neuroscience that completely, totally rewrites this timeline. It details a newly identified temporary brain structure called the arc.
And we are going to track exactly what this structure is, the molecular identity of the cells inside it, and how they travel.
Because this postnatal migration might actually be the secret to high-level human cognition.
Yeah, and it gives us a totally new physical mechanism to understand some of the most complex neurodevelopmental conditions, too.
Right, like we're looking at a direct structural link to things like autism spectrum disorder and early childhood epilepsy.
Exactly.
Because there is this really striking clinical correlation we have to keep in mind for this deep dive. Premature infants who suffer from perinatal hypoxia, so, a lack of oxygen to the brain right around birth, they often face lifelong cognitive challenges.
And by the end of this, we'll see exactly how this newly discovered migration highway might be the specific machinery that gets physically damaged during that vulnerable window.
But to understand the arc, we really have to start with the physical shape of the brain itself, don't we?
We do, yeah. Because you cannot understand this structure without looking at the difference between a smooth brain and a folded brain.
Right, so let's break that down.
So in evolutionary biology, mammalian brains are roughly categorized by their surface topology. You have lissencephalic brains, which are entirely smooth.
Like mice, right.
Mice, yeah, and marmosets are classic examples. And then you have gyrocephalic brains, which are highly folded and wrinkled. Humans, macaques, chimpanzees, sheep, pigs, they all fall into this group.
And that wrinkling or gyrification, that evolved as a physical hack, basically.
Exactly. It's a way to pack more cortical surface area, more actual processing power into the limited volume of a skull.
So the researchers behind this paper, they wanted to see if the deep area right next to the brain's fluid-filled ventricles looked different across these smooth and folded species right at the moment of birth.
And the level of physical evidence they pulled together here is just incredible. I mean, they utilize 3D MRI reconstructions and serial tissue sections treated with a Nissl stain.
A Nissl stain, that's the dye that specifically highlights cell bodies, right?
Right. It allows you to see the exact density and architecture of the cellular landscape. They systematically mapped 6 different species right at their respective times of birth.
What emerged was this massive mathematical correlation.
Yeah, an R value of 0.8291 to be exact. They use something called the gyrification index, which mathematically scores how folded a brain is and mapped it against the size of this region near the ventricle.
So it was nearly 1-to-1.
Essentially, yes.
Yeah.
The deeper the wrinkles and folds in the species brain, the more massive, elaborate, and physically expansive this subventricular structure is.
Wow. So it is literally a direct biological prerequisite for a folded brain. And because this structure has a very distinct curved shape hugging the wall of the lateral ventricle, they named it the arc.
The arc, exactly.
Yeah.
But it isn't just some disorganized blob of extra cells.
Yeah.
It is intensely structured.
Yeah, going through the imaging in the paper, I kind of pictured the arc exactly like a bustling airport terminal, like custom built for immature neurons.
That is a surprisingly perfect analogy. Scientists call these migrating cells neuroblasts. And they identify them by a specific protein marker called double cortin, or DCX.
So if you are a cell expressing DCX, you are a passenger in this airport. And this airport has 4 incredibly distinct tiers.
And those tiers delineate the exact physical progression of a cell preparing to leave the nest.
Let's map it out for everyone. So tier 1 is the ticketing counter. This is a dense, tightly packed line of DCX positive cells pushed right up against the actual wall of the lateral ventricle.
And if you are a mouse or a marmoset with a smooth brain, your airport is basically just a ticketing counter. You don't get anything else.
But in folded brains, like humans and pigs, the architecture extends outward into tier 2. This is the concourse. The cells here break their tight formation, they become more dispersed, spreading deeper into the tissue.
And then tier 3 is where the architecture becomes highly specialized.
Right, this is the food court, or like the VIP lounge. Here, the migrating cells are no longer just floating. They're tightly clustered around a massive network of blood vessels.
Yeah, the researchers used an alpha SMA marker to highlight vascular smooth muscle. And in humans and pigs, the density of blood vessels in this third tier is just staggering.
But in mice, it simply isn't there, right?
Not at all. It's completely absent.
Wow. And finally, you push out into tier 4, the boarding gates. These are dense clusters of cells pointing outward, away from the ventricle, lined up and physically oriented to launch into the deep white matter of the brain.
The airport analogy holds up perfectly when you look at the temporal dynamics, too, because this structure is entirely temporary. It is only packed during the holiday rush, so to speak.
Right. The timeline is wild. The researchers track this in humans. If you look at a fetal brain at 22 gestational weeks, the arc does not exist.
Right. It only begins to structurally organize around 30 weeks, which is exactly when the major folds of the brain are forming.
And it hits its absolute peak volume right at term, right when the baby is born.
Then it undergoes a massive, dramatic decline. By the time a human infant is 7 months old, the arc has severely degraded, particularly those middle concourses and vascular lounges in tiers 2 and 3.
And by age 2, it just vanishes entirely, right?
Completely. The entire structure essentially self-destructs. And they found the exact mirrored timeline in the piglet, forming before birth at embryonic day 100, peaking at day 0, and entirely disappearing by 5 months.
Okay, so let me just do a really quick 5-minute recap here to make sure we're all on the same page.
Good idea.
So bullet point 1. Postnatal migration specifically happens in mammals with folded brains. 2. The arc is a temporary 4-tier transit hub near the ventricles, complete with a vascular VIP lounge. And 3. It completely vanishes by age 2 in humans.
Spot on.
So for you listening, imagine your brain literally reshaping its cellular highways in your first months of life. If you had to guess, what kind of cells require this special postnatal airport?
Well, the transcriptomic data tells us exactly who these passengers are. The cells waiting at those tier 4 boarding gates are primarily GABAergic interneurons.
GABAergic interneurons.
Yeah. In the human arc, almost 80% of them express GAD1, which is the specific genetic marker for these cells. In piglets, it's about 85.5%.
Okay, so they are brakes. Like, if we think of the brain's neural network like a high-performance engine, excitatory neurons are the gas pedal, they trigger actions, they make things fire. GABAergic interneurons are the brakes.
Exactly. They are inhibitory. They calm, regulate, and synchronize the electrical storms in the brain so we don't constantly short circuit.
And those brakes are not manufactured in the arc itself, right?
Now, the arc is purely a staging ground. Their origin story actually begins much earlier in development, deep in the fetal ventral brain, in specialized regions known as the ganglionic eminences, or GEs.
The fetal factories.
I like that, yeah, the factories.
So there are 3 primary factories producing these cells. The medial factory, or MGE, the caudal factory, CGE, and the lateral factory, LGE.
But proving that the infant cells sitting in the postnatal arc actually originated in these specific fetal factories requires some wild technological wizardry.
Oh yeah, the methodology here is intense.
They utilize single-nucleus RNA sequencing, or snRNA-seq. They took human arc tissue from brains right around the time of birth, gestational weeks 30 to 39, and physically isolated 28,550 single nuclei.
28,550, that is wow.
Yeah. And by sequencing the RNA inside each individual nucleus, they're essentially reading the real-time active instruction manual for every single cell.
But just reading the current state wasn't enough for them, was it?
No, because they needed to know the future. So they applied a highly advanced mathematical algorithm called the cell rank Markov chain model.
A Markov chain. Wait, isn't that the same kind of math used to predict complex shifting systems like weather patterns or stock market fluctuations?
Exactly the same math.
So how does an algorithm like that predict the destiny of a biological cell?
Well, a Markov chain calculates the mathematical probability of transitioning from one state to the next based entirely on the specific variables of the current state. In biology, a cell's state is dictated by which genes are turning on and off in any given millisecond.
Okay, I follow.
So by feeding the massive RNA sequencing data into this algorithm, the model identifies the subtle, shifting trajectories of gene expression. It allows researchers to mathematically calculate the fate probability of each cell.
That is so cool. So they aren't just guessing, they're calculating exactly what kind of mature adult neuron each of these infant cells is destined to become.
Right, using math to look into the future of a brain cell.
Yeah.
And the algorithm revealed that these cells retain the specific genetic ID tags of the fetal factories that built them.
Let's do a quick mnemonic map of those markers for the listeners. So if a cell comes from the medial factory, the MGE, it carries specific genetic flags, LHX6 and MAF1, right?
Correct. And the math predicts those will mature into standard regulatory breaks, specifically PV positive or SST positive cells.
Okay. And if it comes from the lateral factory, the LGE, it expresses SP8. And the algorithm says it is almost entirely destined for the olfactory bulb to help process smell.
Yeah. But the really crucial one for our purposes today is the caudal factory, the CGE.
Right. Cells born there carry the TFII and PROX1 markers, and their fate is to become a highly specialized type of break called a VIP-positive interneuron.
This reveals the true purpose of the ARC. It is not indiscriminately flooding the brain with generic inhibitory cells. It is sorting and distributing highly specialized, factory-specific regulatory systems to very precise coordinates in the expanding cortex.
But wait a second. Looking at the source data, something jumped out at me. We said earlier that the piglet is our perfect one-to-one model for the human brain's development here.
It's a very robust model, yes.
But then why does the data say the human arc is made up of about 32% of those MGE medial factory cells, but the piglet arc only has about 9%? Is the pig actually a bad model for human development?
That is an excellent catch. It's an incredible discrepancy, but it doesn't invalidate the model at all. It actually highlights a very specific feature of human evolution.
Oh, really? How so?
Humans are entirely unique in how long we extend our brain development. In humans, that medial factory remains neurogenic, meaning it is actively dividing and producing brand new cells all the way until term.
Oh, wow.
Yeah. In pigs and almost all other mammals, that specific factory shuts down its production lines much earlier in gestation. So that jump from 9% to 32% is a direct molecular footprint of human evolution. We intentionally keep the factory open longer to supply a massive surplus of complex wiring to our uniquely huge frontal cortex.
Okay, that makes perfect sense. Time for our second quick recap. So the arc is packed with diverse inhibitory interneurons, mostly GABAergic breaks.
Right.
They are born in fetal ganglionic eminence factories. We map them using snRNA-seq and Markov chain algorithms, and they use the arc to figure out their final cortical fates.
Spot on again.
So, for you listening, knowing these cells are inhibitory, meaning they calm or regulate brain activity, what might happen if they never reach their destination?
Well, the engine is going to run dangerously hot.
Exactly. So how do they actually get out of the airport and into the deep brain?
To map the physical highways out of the arc, researchers basically had to make a postnatal day zero piglet brain invisible.
Invisible? Seriously?
Yeah, they used a technique called brain tissue clarification combined with 3D light sheet imaging. By rendering the dense fatty tissue transparent, they could specifically illuminate those migrating neuroblasts and physically watch the streams of cells exiting the Tier 4 boarding gates.
That is just incredible technology, and they mapped two totally distinct migratory routes that use completely different physical mechanisms to travel. Let's do another memory trigger. So first, the DICC highway, DICC.
Right, the dorsal stream.
Yeah, the dorsal route heads upward. D for dorsal, I for individual.
Yeah.
These cells are lone rock climbers. They travel completely individually, stretching radially toward the cingulate cortex.
That's the first C.
Right, and they mostly consist of those MGE cells we talked about. To climb, they use a chemical breadcrumb trail. The upper layers of the target cortex secrete a specific molecule called CXCL12.
And the lone climbing cell has a receptor on its surface called CXCR4, that's the second C. It literally sniffs out that chemical trail, climbing the concentration gradient radially through the dense, tangled jungle of the brain to find its exact destination.
It is a chemical homing beacon. Now, contrast that lone climber mechanism with the second route, the VCVT highway. Ventral clumps, VLDLR, temporal.
Yes, the ventral route heads downward toward the temporal cortex. And these cells, mostly the CGE ones, do not travel alone. They travel in massive clumps, forming long, continuous chains.
And they don't climb blindly through the tissue, do they? They ride a biological zip line.
They do. The imaging shows these massive clumps of cells wrapped entirely around specialized glial fibers expressing markers like BLBP and GFAP.
The glial fibers provide a literal physical track.
And to stay together on this track, these clump cells express completely different surface receptors, specifically VLDLR and LRP8, which are part of the renin signaling pathway.
So instead of acting as a chemical nose, these receptors act like molecular Velcro. They allow the cells to physically adhere to one another, forming those tight communal chains so they can navigate the long, incredibly complex downward route together.
And the researchers proved this isn't just a static picture. They proved these cells are actively crawling after birth. They took slices of a day zero piglet brain, kept the tissue alive in a culture dish, and infected the migrating cells with a GFP virus that makes them glow bright fluorescent green.
Over 21 days in vitro, using time-lapse microscopy, you can literally watch the green cells crawling out of the arc.
And the most amazing part, by day 21, the cells hadn't just moved. They had started sprouting complex branching arms called ramified dendrites. They were physically integrating themselves into the existing gray matter network.
That is the definitive proof. Postnatal migration isn't just some lingering cleanup process. It is the active, physical assembly of advanced cortical circuitry happening well after the infant has left the womb.
Absolutely.
Okay, recap number 3. ARC neurons migrate via 2 distinct paths. We have a dorsal route for solitary cells using CXCR4 heading to the cingulate cortex. And we have a ventral route for cellular clumps relying on VLDLR and glial fibers heading to the temporal cortex.
Perfect.
So listener, why would the brain use 2 entirely different modes of transportation, lone wolves versus chained clumps, to build different parts of the cortex?
Well, this is where we hit the true clinical stakes of the paper. We mentioned those specific cells from the caudal factory earlier.
Mm-hmm.
The CGE cells mathematically destined to become VIP-positive interneurons.
Right.
When researchers integrated all this infant tracking data with single-nucleus maps of mature adult human brains, they found something profound. They looked at the exact cortical regions targeted by those upward and downward arc highways, the cingulate cortex and the temporal cortex.
And what did they find?
In adult brains, those specific regions show a massive, dramatic expansion in the density of those VIP-positive breaks.
And we know this expansion is specifically caused by the arc because when they looked at the occipital cortex, the visual processing center at the back of the head, which does not receive any highways from the arc, there is no VIP expansion whatsoever.
Exactly. And in smooth-brained marmosets, which never develop an arc, the expansion doesn't happen anywhere. The arc's sole, specialized purpose is to heavily wire our frontal and temporal lobes with these very specific regulatory breaks.
And those specific lobes, the prefrontal cortex, the anterior cingulate, the temporal cortex, those are the highest order cognitive processing centers in the human brain.
Crucially, they are the exact regions most heavily implicated in autism spectrum disorder, or ASD. Patients with ASD frequently exhibit altered densities of inhibitory interneurons in these exact areas. They also consistently show abnormal gamma oscillations.
Wait, if these VIP cells are breaks, how do they create an oscillation or a brainwave?
Because biological breaks don't just clamp down and stop all electrical activity permanently. They fire in rapid, rhythmic, synchronized pulses. They pulse the breaks on and off, thousands of times a second.
Oh, I see.
That rhythmic pulsing creates high-frequency brainwaves, gamma oscillations. Those specific oscillations are strictly required for focusing attention, retrieving memories, and processing complex sensory information.
So if the brakes aren't installed correctly or there aren't enough of them, the engine's timing is entirely off. The brain can't synchronize those critical gamma waves.
Okay.
And this brings us all the way back to the clinical correlation we started with: premature infants and perinatal hypoxia.
Right. The perinatal window is an incredibly high-risk period for hypoxia ischemia, a sudden lack of blood flow and oxygen to the brain.
And tier 3 of the arc, the VIP lounge where all these cells are staging right at the moment of birth, is entirely defined by being clustered around a massive network of blood vessels.
The arc is critically dependent on that vascular scaffold. If a premature infant suffers perinatal hypoxia, the fragile blood vessels in tier 3 are starved of oxygen and physically damaged.
So if you destroy the infrastructure of the airport concourse, the passengers are permanently grounded. You physically sever the postnatal migratory highways.
The solitary rock climbers heading upward lose their chemical trail. The clumps of commuters heading downward lose their glial zip lines. The cells physically cannot reach the cortex.
That is terrifying.
The catastrophic result is that the frontal and temporal lobes are permanently deprived of their full, necessary complement of VIP-positive interneurons. You create a lifelong structural imbalance between the excitatory gas pedal and the inhibitory brakes in the highest cognitive centers of the brain.
Wow, so this physical mechanism perfectly bridges the gap, explaining exactly how a perinatal brain injury directly leads to the subsequent development of early childhood epilepsy and neurodevelopmental conditions like ASD.
It really does.
Knowing exactly how the machinery breaks is the first crucial step toward eventually figuring out how to protect or repair it. It fundamentally changes how you view a newborn baby. Their brain isn't a finished house. It is an incredibly active, plastic construction zone.
And from an evolutionary perspective, it is an incredibly elegant solution to a profound physical problem. Millions of years ago, the mammalian brain began to fold, creating vast new cortical real estate for higher-order thinking.
But the fetal brain simply ran out of time and physical space to complete the wiring of that new real estate before the baby had to be born.
Exactly. So evolution devised the arc. It is a brilliant temporary scaffolding system designed to ensure that the highly complex folded brains of advanced mammals receive their precise regulatory wiring right at the dawn of postnatal life.
But like any scaffolding, once the walls are up and the wiring is finished, it has to be torn down, which leaves a final, deeply provocative mystery. We know the arc completely self-destructs by the time a child turns 2. But what triggers the demolition?
That is the million-dollar question.
Is there a genetic timer ticking down inside the cells themselves? Does the brain somehow sense that the frontal cortex is fully stocked with interneurons and send a chemical signal back down to the ventricle to shut the factory doors forever?
It is the next great biological puzzle.
So, for you listening, let us know your theories on what triggers the teardown. Because the reality is, we aren't fully built when we leave the womb. We step out into the light, and our brains are still physically crawling, cell by cell, building the exact pathways that allow us to understand the world. Thanks for joining us on this deep dive. Keep questioning the world around you.